Okay, so we're going to talk with this lecture about ATP and anaerobic metabolism. Now, probably 2/3 of this will be review from PE 220, but there's going to be some stuff we did not talk about in 220 that we will talk about here because it's important for exercise. Okay, so to start off, we actually have two of these slides. This is the first one. So we're going to talk about ATP, which is review, and then ATP-PCr system, which is new, or at least new for new content. And then later on, we're going to talk about glycolysis. Okay, so how do we produce energy? And the main way that we produce or really liberate energy is we take ATP, it combines with water, and what happens is we break this ATP down, and it becomes ADP. That's the big part here, right? And what's interesting is that most of the energy that's released is actually lost as heat. Okay, these chemical reactions that are going on that break down ATP are helping to keep your body temperature at 37 degrees Celsius. All right, but some of that, about 20%, 30%, somewhere in there, of that energy that's released, we can then use to perform what we would call mechanical work. So just think about muscle contraction, or your heart contracting, or something else that's going on in your body. Now, the body itself stores very little ATP, and the reminder of this, or the reason for this, is that if we stored all the ATP that we needed to function throughout the day, we would weigh a lot. It would just be, it's very inefficient to do that. And so as a result, we don't store a lot of ATP. Instead, what we do is we take ADP, we form ATP, that ATP is broken back down into ADP, and so on and so forth. And so as a result, since we don't store a lot of ATP, we need to be able to produce it in times when we need energy. Okay, for example, exercise. All right, so this leads to the first of several energy systems that we're going to talk about. And the first one is what we call the ATP-PCr, ATP phosphocreatine system. Okay, and so I want you to just get a basic overview of how this works, and then as well as, well, when is this system being used, or when is it being used a lot? And then how can we inhibit this system? So to start off, we've got ATP-PCr, so phosphocreatine. Okay, so here is my ATP, which is the end product here, and then we've got this stuff called phosphocreatine. Okay, like I said a minute ago, we don't store very much ATP in our cells, but we do store, one of the things we do store as well is something called phosphocreatine. And so what can happen is we can take this phosphocreatine and we can break it down with the help of an enzyme called CK, or creatine kinase. Okay, so just to circle here, this is that. All right, and so what happens is we break this phosphocreatine down and three things are produced. We've got creatine, we've got a phosphate group, which is important, and then we've got this energy. Okay, and then what happens is we can take that energy that's been released from phosphocreatine to take an inorganic phosphate and an ADP to form ATP. Now, this is something that occurs very quickly. Okay, really, whenever you begin to do something, okay, so the start of exercise, you're primarily using your ATP-PCr system. Okay, so it's very fast acting, but the problem is it doesn't last for very long. Okay, so generally 10 to 15 seconds, you don't have very much phosphocreatine left, and so you can't rely on that system anymore. Okay, so 10, 15 seconds, think 100-meter dash. Unless you're like Usain Bolt, you've probably depleted your PCR in that 100 meters. He'd be able to go almost 200 in that time point. And so that's why we don't just rely on the ATP-PCr system. Now, creatine kinase is this enzyme, right? And this enzyme helps this reaction to occur. But what's important to know is your body is very efficient, okay? And so it's only going to form ATP if you've got low levels of ATP and or high levels of ADP. Okay, if you've got high levels of ADP, that activates or that helps increase the activity of creatine kinase so you can form ATP. But if you've got lots of ATP floating around, just kind of hanging out there, that's an inhibitory signal. Okay, and so the creatine kinase doesn't work as efficiently and you don't form more ATP because we're not trying to store ATP. All right, so this is an example of a negative feedback loop. Lots of ADP causes creatine kinase to form ATP. If I'm not using that ATP, that's going to inhibit creatine kinase. Okay, and then we're going to go to glycolysis. And so this is the second of these learning topic and outcome slides. Okay, and this is mostly review. There's going to be a few things that we add into this. So we've got glycolysis, and this diagram actually shows you glycolysis. Now, if you take 220 from me, we don't dive this deep into glycolysis, right? So I don't have you remember that glucose turns to glucose 6-phosphate, turns into fructose 6-phosphate, yada, yada, yada. All I have you remember is that 1 glucose turns into 2-pyruvate. Okay, so pyruvate times 2. Okay, we're going to dive a little bit deeper. But a couple things I want you to remember is that this occurs in the cytosol of the cell. It's the egg white. Okay, it starts with glucose. And remember, this first half costs 2 ATP. And those 2 ATP are right here and right here. Okay, but then the second half, we produce 4 ATP. And so those ATP are formed here and here. Now remember, this splits at step 5, we actually end up with 2 molecules, which ends up having 2 pyruvate. So we end up with 2 ATP here. So 2, that's a terrible looking 2. And then we get 2 ATP here. So we end up netting 4 ATP, right? We're at -2 halfway through, and then we gain 4 back, so we're +2. Glycolysis doesn't require oxygen, but it does produce a molecule called NADH, which is going to be very important when it comes to the electron transport chain. And so here we see this NADH, and again, there's 2 of these that are formed because we've formed this fructose 1,6-bisphosphate splits into 2. All right. So as a result, we've got 2-pyruvate, but this only happens if oxygen is available. If no oxygen is available, then we form 2 lactate molecules instead. Now with glycolysis, this is the primary system that occurs for events that last 1 to 2 minutes. Okay, so with ATP-PCr, that's like your 100-meter dash, okay, if we're talking elite athletes. All right, when it comes to the glycolysis, this is the next 2 minutes, okay? And so this is like when you see somebody at track running an 800 meters. They're relying almost solely on glycolysis. It's after this 800 where we start getting into the 1500 where we start getting into that aerobic metabolism. Now, just like creatine kinase is the limiting enzyme because it's the only enzyme, well, glycolysis has a rate-limiting enzyme as well. And it's called phosphofructokinase, which is right here. So it's the third enzyme. Now, the reason that it does, or that it is the rate-limiting enzyme, is because it's extremely sensitive to the concentrations of ATP, ADP, and phosphate. And just like creatine kinase, if we have high levels of ADP and or phosphate, then phosphofructokinase, PFK, will have high activity levels. But if I've got lots of ATP floating around, phosphofructokinase decreases its activity. Again, a negative feedback loop. And so that's where we're going to end right now. And this is essentially the anaerobic part of metabolism.